The discovery that DNA contains our genetic information implied that there was a code, some sort of a method to translate the sequence of DNA, written in 4 letters, G, A, T, and C, into the 20 amino acid letters of proteins. RNA was suspected to be the go-between, bridging DNA and protein synthesis. But how? Deciphering the code and how it worked was achieved through systematic experimentation, predominantly performed by Marshall Nierenberg and his team at the National Institutes of Health. This occurred from 1958 to 1962. Nierenberg hypothesized that RNA could in some fashion dictate the production of a specific protein. In order to test this idea, he synthesized artificial RNA. He then added this synthetic message to a reaction mixture, a solution that would be able to use the RNA and produce whatever protein it may correspond to. The reaction mixture was obtained by breaking open E. coli bacteria and collecting the contents that had ribosomes and all the goodies needed to produce proteins. The first RNA tested was the simple polymer of uracil, just a long RNA molecule made up of one uracil bound to another. Adding this poly-U to the reaction mixture suddenly produced a protein that was made only of one type of amino acid, phenylalanine. Polyuracil, poly-U, produced polyphenylalanine. These results were truly amazing. Nierenberg had discovered that RNA could dictate the production of a specific protein. It wasn't long before the team could show that poly-A corresponded to polylysine, poly-G to polyglycine, and poly-C to polyproline. Ultimately, the code of all 20 amino acids was deciphered. It turned out that the genetic code was made of 3-letter coding units called codons. In view of the fact that RNA has 4 possible letters, the nucleotide bases G, A, U, and C, a total of 64 codons are possible, 4 to the power of 3. These 64 codons code for the 20 amino acids of proteins, as is shown in the table of the genetic code shown right here. Some amino acids have only one codon, such as methionine and tryptophan, while leucine, arginine, and serine have 6 different codons each. In addition, 3 codons serve as stop codons, instructing the ribosome where to stop protein synthesis. Each codon has 3 bases. One can derive the codon from the table for any amino acid by determining the correct base for the first, second, and third position of the codon. Thus, for example, the codon for methionine, AUG, is A at the first position, U at position 2, and G in the third position. Similarly, UGG codes for tryptophan. You certainly do not need to learn the codes for all 20 amino acids. You should, however, remember how to extract the codon from the genetic code table for any given amino acid. 2 more aspects of the code still must be considered. The first is the directionality of the codon itself. RNA is a continuous linear polymer, starting at its 5' end. The directionality of the RNA and the genetic code are very important. Reading the codon AUG, for example, starts at the A at the 5' end, the first position, and thus correctly gives the instruction for methionine. However, mistakenly, reading AUG from the opposite end, that is GUA, this would lead to an error, ultimately incorporating valine instead of methionine. Thus, it is important to remember that codons are directional and are to be read starting at the 5' first position. The second point is to establish where to start reading, or where to initiate translation and protein synthesis. Consider the following RNA sequence: ACC, GAG, CUU, and so forth. If codons are read starting with ACC, then the corresponding amino acid sequence would be T, E, and so on. If, however, we were to begin reading at position 2, at C, and reading triplet codons CCG, AGC, then the protein that would be produced would be totally different. The initial point where one should start reading the codons is referred to as the correct reading frame. Obviously, RNA can be read in 3 different reading frames, and only one would actually correspond to the correct and intended protein. Thus, it is critical to identify the correct reading frame. Identification of the correct reading frame is also a unique function of the ribosomes. So you see, ribosomes are amazing molecular machines that recognize messenger RNA, identify the reading frame, and are able to translate the message coded for in RNA to implement the production of the proteins of our cells.